For today's learning target, we will be going over learning target 6.5. I can identify sources of genetic variation in sexual reproduction. At this point, you should have already answered the launch questions on your exit ticket and logged your percentage score on the Mastery Self Tracker. If you haven't done so already, pause the video to make sure you do this. For today's agenda, we will have a few announcements, and then you will take notes on your notes handout on genetic variation for the learning target 6.5. After you finish taking notes using this PowerPoint and the video, you will continue with independent practice on classwork. If you didn't finish the classwork and the note-taking skills on the video from yesterday, you will also be finishing that. Some announcements are due tomorrow is your weekly homework packet 27. You will also have a quiz 27 on sexual reproduction, the topic that we have been working on since Monday. Tomorrow is also the last day to turn in the weekly homework packet 26 from last week. Tomorrow will also be the last day to make up quiz 26 on chromosomes from last week. The focus question for today is, how does sexual reproduction lead to genetic variation? Hopefully, by the end of this presentation, at the end of the instructional period, you will at least be able to identify 3 different ways that sexual reproduction leads to genetic variation. As a review of what we've been doing in class since Monday, go ahead and pause this video to complete the chart below. This chart compares the processes of mitosis and meiosis. Hopefully, you've taken the time to pause the video and complete the chart below for review. Remember that in mitosis, at the end of the process, you should have identical cells. At the end of meiosis, you would end up with different cells. They would be random. At the end of mitosis, you would produce 2 cells. On the other hand, meiosis produces 4 different cells. Mitosis is used as a process to duplicate or replicate somatic cells. These are body cells. On the other hand, meiosis produces gametes, or in other words, sex cells. At the end of mitosis, the cellular process produces diploid cells. This is indicated with 2N for 2 sets of chromosomes. For meiosis, the cellular process results in haploid cells, which is indicated with the letter N for 1 set of chromosome. And finally, the purpose of mitosis is for cell growth and repair, whereas meiosis is for sexual reproduction. Go ahead and make sure that your table or your chart is accurately filled out. Sexual reproduction is important because it results in infinite possibilities of genetic variation. In other words, sexual reproduction results in offspring that are each genetically unique. They differ from both parents and also from each other. This occurs for a number of reasons, and I'll list out 4 of them. The first reason is as a result of independent assortment of chromosomes during meiosis. The second reason is a result of genetic recombination or crossing over that also happens in meiosis. And the third and fourth reasons are 1. Random gamete formation, and 2. Random fertilization of gametes. We will go over all 4 ways of how sexual reproduction leads to genetic variation in species. The first way that sexual reproduction leads to genetic variation is independent assortment. Essentially, when cells divide during meiosis, homologous chromosomes are randomly distributed to daughter cells, and different chromosomes segregate independently of each other. This is called independent assortment. It happens during metaphase 1 of meiosis and results in gametes that have unique combinations of chromosomes. The second way that sexual reproduction leads to genetic variation is crossing over. Crossing over during meiosis increases genetic diversity. When homologous chromosomes form pairs during prophase 1 of meiosis 1, crossing over can occur. It is essentially the exchange of genetic material or chromosome segments between homologous chromosomes. This results in new combinations of genes on each chromosome. This diagram right here represents how crossing over results in the exchange of DNA between homologous chromosomes. In step 1, and this happens during prophase 1 in meiosis, two homologous chromosomes pair up with each other. The second thing that happens during a crossing over scene is that the chromatids are very close to each other and the segments actually cross. At this point, going on to the third step, is that the segments break off of the chromatids and they reattach to the other homologous chromosome. And so as you can see in the third and final picture, the chromosomes actually include two different colors indicating that the DNA has been exchanged between the homologous chromosomes. Before we move forward to discuss the formation of gametes, we'll engage in a unique and brief activity to help clarify how the gametes are formed. Your teacher should have passed out a penny or a coin to represent alleles parents pass on to the offspring at your table. You will be using the last page of this notes handout to write down the alleles that you inherit from either your mom or your dad. You will flip the penny or coin at least 23 times, and when you land on a heads, this indicates that you inherited an allele from your mom. Use one color to indicate this on the last page of your notes handout. If you land on a tails, this means that you are inheriting an allele from your dad, and you may also indicate this with a different color on the last page of your notes handout. After you do this for chromosome 1 to chromosome 23, make sure to compare your own gamete that you formed with your partner at your table. Are they the same or are they unique? Lastly, check with Ms. Fox if you have the same formed gamete as she does. If you have the exact same gamete that she made, you can get $10. The activity before this actually shows how humans have over 8 million configurations for the unique haploid cells. For each chromosome, it's random whether the organism inherits the version from either the mom or the dad. And because we have one set of 23 chromosomes and we flipped the coin at least 23 times, that makes us 2 to the power of 23, which means that there are 8,388,608 unique gametes that are possible. It is the specific processes of meiosis resulting in 4 unique haploid cells that result in these many combinations. This independent assortment in which the chromosome inherited from either the father or the mother can sort into any gamete and produces the potential for tremendous genetic variation. Finally, the final way that sexual reproduction leads to genetic variation is through fertilization. Again, the number of different kinds of sperm or eggs, or in other words, the gametes, that can be produced in a human species is 2 to the power of 23. When an egg and sperm unite to create a zygote, the number of possible genetic variations that can result from that is actually the answer when you multiply 2 to the power of 23 by also 2 to the power of 23. Essentially, that's over 70 trillion different zygotes that could be formed during sexual reproduction in humans. At the end of meiosis, 4 haploid cells have essentially been produced. However, the cells are not yet gametes. The cells need to develop before they become mature gametes capable of fertilization. As a result, we'll go over 2 processes that allow the cells to develop into sperm and eggs. These are spermatogenesis and oogenesis. Spermatogenesis is the process by which sperm are produced in male animals. This occurs in the testes. Essentially, what is happening is that primary spermatocytes go through the first cell division of meiosis to produce secondary spermatocytes. This is seen in the diagram to the right here. These are haploid cells. Secondary spermatocytes then quickly complete the meiotic division to become spermatids, as shown in this row, which are also haploid cells. The 4 haploid cells produced from meiosis develop a flagellum tail and compact headpiece to become mature sperm cells, which are capable of swimming and fertilizing an egg. The compact head, which has lost most of its cytoplasm, is key in the formation of a streamlined shape. Finally, the middle piece of the sperm connecting the head to the tail contains mitochondria, providing energy to the cell. The sperm cell essentially contributes only DNA to the zygote. On the other hand, the other process that occurs before fertilization is oogenesis. This is the process by which gametes are produced in female animals, and this occurs in the ovaries. During cytokinesis following meiosis I, the cytoplasm divides unequally. The cell receiving almost all the cytoplasm will become the ovum, and the other smaller cells are called polar bodies and will not survive. The next slide will indicate a diagram of oogenesis, which I will walk through. This diagram indicates the process of oogenesis. Oogenesis begins before birth and is not completed until after fertilization. Oogenesis begins when oogonia, which are the immature eggs that form in the ovaries before birth and have the diploid number of chromosomes, undergo mitosis to form primary oocytes, also with a diploid number of 2n. Oogenesis proceeds as a primary oocyte undergoes the first cell division of meiosis to form secondary oocyte with the haploid number of chromosomes. A secondary oocyte only undergoes the second meiotic cell division to form a haploid ovum if it is fertilized by a sperm. This means that a sperm needs to swim up to the egg in order to be fertilized. The one egg cell that results from meiosis contains most of the cytoplasm, nutrients, and organelles. This unequal distribution of materials produces one large cell, as indicated at the very end here. This is the mature ovum. This contains all the cytoplasm, nutrients, and organelles. All the other 3 cells produced here, we call them polar bodies, which eventually break down. The larger cell here undergoes meiosis II, once again producing a large cell in a polar body. The large cell develops into the mature gamete called an ovum.